fuse it with an activator or a repressor domain, and the cool thing for the TET system, I think it's, do we have a small molecule ligand, and this can influence if the protein binds to the DNA or not, and so you can decide if you switch gene expression on and off, and the most of you will know the system, have already heard about it or already applied it. And but there are some cons, and it's worth to look for alternatives. The cons are, there are strong positioning effects. If you insert this TET system very often close by, silencing systems do influence your regulation. You have a certain metabolic burden, so it's always good to think about alternatives. And yeah, several years ago, or in the meantime, I think it's 20 years ago, I thought, okay, maybe I find an alternative to this classical TET on, TET off system. And yeah, the idea was, can we find or select an RNA aptamers that somehow influence the system? And you can, in principle, think of different ideas. You can think about as an aptamer as alternative inducer, here shown on the right side, where the aptamer goes into the tetracycline binding pocket and leads to this this conformational switches that are changes in the proteins that normally is triggered by tetracycline. Or you also could think that the RNA, yeah, somehow interferes with the DNA binding. So this was the attention at this time. Yeah, it was 2002, I think. Yeah, to look for an RNA as an alternative inducer or competitor. And finally, this also worked out. An aptamer was selected at this time by Günter Meyer when he was at a company Nascacell. Then we performed an in vivo screening, and we came up here with this little guy shown on the left side. This is an RNA aptamer, and this aptamer could indeed really activate TET control gene expression. Here was our system at this time. We worked in E. coli, in bacteria, with 2 plasmids, and from this left plasmid here, TET-R was expressed. On the genome, we had a reporter that was under control of tetracycline, and of course you can add of the TET-R, you can add tetracycline to displace the protein from the DNA. But in our case, we expressed on a second plasmid our RNA aptamer, and this aptamer also leads to the induction of the TET-R control gene expression. As you can see here, the aptamer is induced by IBTG, so it was a TAC promoter. And if we increase the amount of IBTG, this means if we increase the amount of aptamer in the cells, we can switch on gene expression. Gray and black here are different expression level of the aptamers. Yeah, so this was a cool story in the beginning. And then we were interested in how this TET-R RNA, TET-R DNA interface looks like. And to our surprise, the binding of the TET-R of the protein to the RNA was as efficient as the DNA. So yeah, on the left side is the RNA binding, on the left side RNA binding, right side DNA binding, and the KD was nearly the same. And here with this band shift, you can see that if you have the aptamer, then you can nicely shift it in the presence of protein, and it's nearly the same efficiency as with the DNA, with the TET operator. Yeah, if you compare left the RNA, right the DNA. And another cool thing was that there is a competition between DNA and RNA. So if you have here your DNA and you add the protein, then you can shift the DNA. And then if you add increasing amounts of aptamer, then you can compete with this DNA binding of TET-R. So the RNA can displace TET-R from the DNA and vice versa. Also the DNA, you can do these experiments, vice versa. The DNA can also display the protein from the RNA. And finally, and this was really cool, if you have TET-R, if you have doxycycline, or if you add doxycycline or tetracycline, then it leads to a conformational change within the protein, and this leads to the dissociation of TET-R from the DNA, and the same occurred with RNA. So you can use this ligand of the protein to displace the protein, not only from the TET operator, but also from our RNA. And then we thought, okay, this further biochemical characterization, we did a lot of genetic and biochemical analysis. And here we could prove the secondary structure. It's an inline probing gel where you can distinguish between more flexible and more restricted bases. And what you can see here is that you have a certain flexibility in this middle part here of the aptamer. And if you go from the left to the right, increasing amounts of the protein, then you see that this flexibilities, these signals disappear, and this means that you have a stabilization, and this is an indication that the protein really binds here in this middle part, and we could completely exchange the upper and the lower helix. So these helices are a kind of scaffold for our RNA aptamer, and the binding site for the protein is here in the middle. Good. Then we thought, okay, it really worked nice in bacteria. We could here, as shown, nicely activate gene expression. So we had an alternative activator for TET-R, and then we thought, okay, principle, it's a cool device, and what else can we do with this? What can we, how can we apply this aptamer for other gene regulatory purposes? And then here it's finally a summary of what we did with this little guy. You can think about inserting this aptamer in the untranslated region of a messenger RNA of your gene of interest. And then if the protein binds, it's kind of roadblock, and the ribosome, which recognize here the cap structure and then normally scans through this 5' UTR. It's somehow then inhibited by this roadblock here. So you switch off gene expression. And if you add tetracycline, it leads to this displacement from the RNA, as I mentioned earlier. And then the ribosome can read through. So this is one idea. Another idea is if you add this aptamer close to a 5' splice site, then you may interfere with the ribosomal assembly. So you may then have an block of splicing of the constitutive splicing. And another idea we had was that we can interfere with microRNA processing by replacing some parts of the precursor with our aptamer. And I want to give you some examples if this worked out. And here is, as a first example, the control of this microRNA processing. You know, microRNAs are transcribed as long precursors in the nucleus. We have a first endonucleolytic cleavage by pressure. You have the pre-microRNAs, they are exported, and then in the cytoplasm there's a second nuclease, this is dicer, and this leads to this small 22 nucleotide long double strands that are then loaded into the RISC complex, and the RISC complex then acts on the target mRNA. So it's very simplified, and if you are in the field, you know that there are many more bypass and auxiliary proteins. However, this cleavage by dicer is a very crucial step, and for this you need this pre-RNAs. This is this double-stranded part here, where you have the red part will be the mature microRNA, and then you always have a kind of terminal loop, and this terminal loop we exchanged with our TET-R aptamer. And the idea was then, if the TET repressor binds, then it may be simply a steric hindrance for dicer. If we add doxycycline, if the protein dissociates from this aptamer, then dicer may cleave and lead to mature microRNAs. And this is one example for a microRNA 199. We did it in total for, I think, 3 or 4 different microRNAs, and we determined the amount of mature microRNAs by qRT-PCR. And what we could show is that you have here the microRNAs detectable in the construct where we have the precursor topped with our aptamer. And then if we express TET-R, this is the second line here, or this line here, then we can nearly completely prevent the formation of the mature microRNA. If we then add, in addition to TET-R doxycycline, then we can restore the level of microRNAs. And this worked excellent for 199, but also for the other microRNAs we tested. And as a next step, then we had a reporter assay, luciferase, and in the 3' UTR, we cloned the perfect match of our microRNA's target site. And this means if microRNA is produced, it will bind here to this perfect match and shut off gene expression. And if we add TET-R, then we prevent the microRNA formation. And we can see here that we have an increase in gene expression. And again, we can revert it or can switch it back if we add doxycycline. So this system really works fine, and we were quite happy. And then we thought about the other potential targets. And we cloned the aptamer and the 5' UTR, and you can see here exemplarily how it works. You have gene expression without the protein. If you co-express the protein, you can switch off gene expression. If you add doxycycline, you can again switch on gene expression. And in any case, or in every case, we have nice dose-dependent regulation if we add doxycycline. And we have here, of course, a dose-dependent effect also from the copy number, so we can increase translation regulation if we add not only 1 but 2 or 3 of this aptamers in the 5' UTR. And we then also showed that we can control the constitutive splicing if we have the aptamer close to the 5' splice site. So here, if you think about applying this system, you should have a look into this publication, because we really did a lot of optimization here. It's the distance from the splice site, and the stability of the aptamer is crucial for efficient regulation, but in principle it works. And you also can combine the splice site regulation with translation regulation to further increase the dynamic range of regulation. Yeah, and finally, we were interested, of course, how our little guy looks like, and together with Eve Müller in Erlangen, it was possible to solve the crystal structure of the co-crystal. And here on the left side, you see that TET-R is a homodimer. It has 10 helices in each monomer, and marked in red are the helix-turn-helix motifs. So these are the two helices which are responsible for the very specific recognition of the DNA, here shown in yellow. And in orange, we have now here our aptamer, and it binds exactly to the same sites, to the same helix-turn-helix motif as as a DNA. And if you have here a look to this shape, it looks a little bit like DNA mimicry. Do I have here on the left side and on the right side these helical parts, which are the scaffold of our RNA. And here this middle, this internal bunch, this internal asymmetric loop folds, maybe something which looks similar like this inner part here of the DNA. And very important is that we really have the same amino acids, the lysine 42 and glutamine 38, that are recognized by the from the RNA aptamer, or that make the specific contacts, and these are exactly the same positions in the proteins that also contact the DNA. And then we observe the following. If you have a look here, then you see a CHE, and here the HE is marked in red, and CHE is a perfect 3' splice site. And then came in our mind, maybe we can use this as an alternative 3' splice site, so that we can, depending on the presence of TET-R, shift between a canonical and an alternative splice site. And we thought that this may be a perfect site. If you see here, then you have direct contacts of the helix-to-helix motif here in dark blue to these nucleotides, and at least we thought that if the protein binds, then these two positions, A7 and G8, are masked by the protein. And the idea was the following. We have a GFP reporter gene, and we divided this GFP directly after the ADG. So we inserted a first intron, we inserted an intron between the ATG, between the start codon and the GFP coding region. And we have here, this AG is the canonical, the normal 3' splice site. And then here, exemplarily shown is this CAG, which could be an alternative splice site, yeah? And then if it works, then the idea would be that without TET-R, this splice site here is used, and we end up with an ADG and a GFP, so with the correct message. And if this one is is used, then we have splicing between here and here. So this means we have an ATG, and then we have GFP with additional amino acids, the sequence here translated into amino acids. So some additional amino acids and additional information at the end terminus of the protein. Yeah, and did it work? Yes, it works perfectly fine. We have here the usage of the one splice site. If we add the protein, we can nearly completely shift it to the other splice site. And if we add doxycycline, we can revert this effect. But if you have a closer look, then you see that this is nice regulation, but the regulation is exactly vice versa as we expected. This splice site here is only used if TET-R is present. And if no TET-R is present, the canonical ones is used. If we add TET, then this one is used. If we add doxycycline and displace TET-R, again, this one is used. Yeah, we can nicely shift between here and here and here, but in exactly the opposite direction. So actually, we have no clue why it is in this case. Maybe somehow TET-R recruits some splicing factors that accelerate, or here this GA is maybe in the free form extremely fixed. Maybe the free form of the aptamer looks different, and the GA and the A7 is somehow stabilized within the aptamer structure. And if you have here a look into the electron density, then you see that the hydrogen bond between this GA and the protein is not as stable, so there may be a certain flexibility. So currently these are pure speculations, but we thought, okay, it does not regulate in the direction we originally thought, but it regulates, and we want to use this. And the idea was now, as I mentioned, this upper helix here, it's simply a scaffold, and you can exchange it in everything you want. And so we cut it here, and we exchange this upper helical part of our aptamer into a nuclear localization sequence. And the idea then is that if we have this splice site used, then we have a normal GFP with the ATG. And if we have this splice site used, then we attach this nuclear localization sequence to this GFP. So we have here a switch between two isoforms, and one isoform is tagged by the sequence of our choice. And this indeed worked perfectly. We have here of the one side, this situation where we have ATG and GFP. We had to elongate GFP with a cytoplasmic protein. So GFP is too small, it goes into the nucleus by itself. But if you attach a larger protein, then you can keep this in the nucleus. As you can see here, all the GFP is in the nucleus. And then if we add TET-R, then we change the splice site from here to here, and we attach this nuclear localization sequence to our message. And then this completely moves TET-R with GFP into the nucleus. Yeah, if you distinguish between here and here. And then if you add doxycycline, you can revert this effect, and you can find doxycycline back, the GFP back in the nucleus. So this worked excellent, and we hope that many people see this and then may apply our system, because in principle you not only can work with the with the nuclear localization sequence, in principle, you can attach any tag you want to your protein of interest. Yeah, and with this, I'm at the end. I talked today about a TET-R aptamer, and we think it's a nice tool. It's a nice protein-based RNA switch. We have this RNA aptamer that binds to TET-R. The TET-R bind the RNA aptamer with the same or with a similar affinity as the DNA, and they both compete. So you can use RNA to destroy the DNA binding, and you can use DNA to destroy the RNA binding. Yeah, so you can, doxycycline can displace TET-R from both the RNA and the DNA. And so we can use this to activate gene expression in bacteria, to control translation initiation, to control constitutive splicing, or also alternative splicing. And with this, I'm at the end. I want to thank, of course, all my lab members for their many contributions, and you for your attention.